So yeah, lack of words. It's pronounced like stock and stock market. So I have the pleasure to go more into detail about the functional types of connectivity and how we can use them to make statements about segregation in the human brain, but also the monkey brain. And one of the things that I think we have not used to motivate why brain parcellation can be interesting is the following kind of meta-level distinction. It's not entirely clear what are the most relevant organizational principles in the brain in general, but two of the things that we used over more than 100 years now to distinguish these are what you see on the left, functional segregation, and on the right, functional integration. So functional segregation, which is perhaps a little closer to connectivity-based parcellation, departs from the idea that the brain, as mapped out by Brodmann and the folks, probably has territories or compartments, and each of these harbors a set of neuronal populations that subserve distinct classes of neural processing, and this is how kind of functional specificity comes into the brain. Yeah? So the emphasis is on particular parts of the brain subserve particular types of function. On the other end of the extreme, we have functional integration that takes a different focus by saying cognitive function may really be a manifestation of various functionally specialized parts of the brain working together over short range and long range connections. So, and I say continuum because these two extreme views are probably inextricable. So how can you really do functional integration between distant brain regions if those are not functionally specialized? How would that be interesting? So how did we actually get there? You already heard from Paula that a gold standard method that started in the 70s is still very interesting today is axonal tracing. So, and the cat and the monkey were two species that we really used a lot to make these axonal tracing experiments in the anterograde and retrograde directions. So, and if you do this in the cat, which is shown here, we made a very important observation that actually is a foundational feature of the brain that actually makes brain parcellation possible, and that is the following. So if you look at this here, we have various brain regions twice, once on the x-axis, once on the y-axis. And you can see that each particular part of the brain has a unique set of input and output connections. Okay? So and this is something that we believe to be true in brain biology in general. And this is why it makes sense to put it on its head and ask, how is functional connectivity to the rest of the brain different to identify candidate boundaries between brain regions? Okay? So it really comes from animal work that we today do brain parcellation and brain imaging. We heard already there are different types of functional connectivity that you can use to do brain parcellation. So a typical question that people who get into this space of brain imaging methodology have is, why should I use a particular type of functional connectivity rather than another one? So let's say you're working in a lab and you have equal access to the fusion MRI, resting state functional connectivity, and meta-analytic connectivity modeling. Which one should you choose? So I think first of all it's interesting or important to say that there are a lot of researchers' degrees of freedom in brain parcellation in general, so there are many knobs to twiddle and many choices to make. And I think it is important to really keep in mind what kind of neurobiological question or neuroscientific kind of goal do I actually pursue? And that really nudges a lot of these choices into a direction. Yeah? So it doesn't give definitive answers, but by really keeping in mind what is the type of statement that I would like to make about the brain, that really helps in making some of these choices, such as this one here. So all of these three have different advantages and disadvantages that may more or less come into play in your particular type of question. So we already heard a lot of the kind of disadvantages of diffusion in the previous talks, but it also has advantages, because if you ask, which type of functional connectivity is actually closest to these axonal fiber tracts where kind of we came up with the idea that a particular type of brain region might have a unique set of connectivity to the rest of the brain, then probably diffusion MRI is closest. So it's important to keep in mind that you cannot isolate single axons, so it's much more limited in that respect. And this is why tracing in monkeys is still a very important method today. But if you really want to make statements that are close to neuroanatomy in humans, then you're probably still, even given all the disadvantages that it has, you're probably still well off using this type of connectivity modality. So, and I would also actually say that probably the highest number of brain parcellation papers in brain imaging are probably still based on diffusion. So this is how it started, like in 2003-04. And then we saw always more resting state functional connectivity-based brain parcellations. So, and for those who are not familiar with the resting state, the important aspect and how it's different from task-related functional connectivity is that people are just in a scanner supposed to do nothing in particular. So now we could argue, is this a particular type of task or is this no task? The important difference is that when you measure this type of bold signal or this type of bold signal, here you don't have a experimentally motivated, well-defined paradigm, as psychologists and experimentalists call it, where you try to encourage recruitment of certain classes of cognitive processes rather than kind of just let the mind go. Okay? So the difference is, this is kind of like how different parts of the brain talk to each other while people follow a structured task, whereas in resting state, you don't have that feature. Okay? What else? I think it is easier to acquire resting state data at scale. So in a lot of these general purpose open data sets, oftentimes you have T1 structural data and you have resting state, but it's less common that you also have diffusion. And meta-analytic connectivity modeling just depends on experiments. So you incur a lot of additional biases, and it just takes more effort to really acquire these types of data. So, for example, the databases that are used to come up with this task-dependent type of functional connectivity, they are kind of like vulnerable to interests in science. Yeah? So in the BrainMap database, for example, we just have a lot of studies on social affective paradigms because it's just interesting to many people to investigate social affective processing. But there are like relatively less studies, for example, on visceral processing or visceral motor. So if you're investigating brain regions that are related to the less well-represented studies in the fMRI test databases that you need, then you may incur certain biases. Okay? So long story short, each of those has different advantages and disadvantages, and whether or not you should choose one rather than another should probably be driven by what your ultimate goal of the study is. All right. So there are a lot of different types of brain parcellation, and there's some kind of overarching principles that seem to be emerging. I will just mention some of them. Regarding boundaries, as far as I know, the higher you go up the neural processing hierarchy, so away from motor cortices, sensory cortices, to like more frontal areas, I think there are some indicators that the boundaries may get fuzzier as you move up the neural processing hierarchy. So, and another thing that may or may not be connected to this observation is, as you go up the neural processing hierarchy, there's also the tendency that a particular brain region is connected to a diversity of other systems across the brain. Okay? So things like, if you think in the world of ICA brain networks, saliency network, default mode network, and so on, as you go up the neural processing hierarchy, a particular brain region is likely to have connections to always more of these systems at the same time. Okay? Just to give one particular example, the medial prefrontal cortex is a favorite brain region of social neuroscientists, for example. And already in monkey studies in the 80s, it was described in cytoarchitecture that the transition between the anterior cingulate cortex and B810 medial prefrontal cortex, so they're different lobes. Yeah? One is frontal lobe, the other one is cingulate lobe. So textbook knowledge would be there should be a pretty serious boundary between those. It has already been observed under the microscope that the change in the properties of the neurons is actually very gradual. Okay? So this is, of course, reflected and confirmed by what we've seen so far in brain parcellation. All right. So let me just go through the process. Some questions we're going into kind of like, how do we actually choose the region of interest? And there's really a diversity of ways in which you can come up with a starting region. So first of all, a region of interest is usually the same as a volume of interest. There's just a difference in whether you may be working in surface or not. But VOI and ROI, if some people have ever been wondering, at least in the brain parcellation literature, it's usually the same. So how you define this should also be driven by what you ultimately want to say. Yeah? So let's say you want to make statements about that there are perhaps different functional areas that may subserve different cognitive processes in this part of the brain. Then perhaps you have more reason to choose a region of interest that you extracted perhaps from a fMRI contrast, from an experiment, for example. Whereas if you want to make statements about Brodmann area instead, it may perhaps be more convincing if you choose a region of interest that has a more neuroanatomical motivation, such as the Jülich histological address or something. Okay? So, and this is another example how these many choices that an investigator has to make going through a connectivity-based parcellation analysis, how you can kind of like nudge those and align them with what statements you ultimately want to make. So you see that in the case of meta-analytic connectivity modeling, so the experiment-based type of functional connectivity, the way it works is the following. You would browse through the database and find various types of experiments that show at least a focus of activation in this region of interest. So, and the most common question that people ask in this situation is, how do you know which studies to choose? So if you're interested in, let's say, language, why don't you just choose language studies in the database, which is typically not what is done? And a possible answer is because you don't really know what the mapping from brain to cognitive functions is. Yeah? So if we really knew the ground truth on how various parts of the mind are related to each other and how they map to phenomena in the brain, then we could perhaps subselect those, but like language also probably plays a role in various processes that we may not call language in certain experiments, for example. So typically you just take any experiment that has one focus of neural activity change in your region of interest. Okay? So you end up with a subset of the studies, and those help you, and I'll go into detail how this works, to parcellate your region of interest into disjoint areas. And oftentimes, that is actually not the end, just to foreshadow this, of the connectivity-based parcellation. Typically, you want to kind of like post hoc investigate what these different parcels may actually mean. And two very common ways to do this is to perform another connectivity analysis, which is different from this one, because here we do voxel-based brain parcellation, whereas here you would, with the parcels that you already obtained, use those as seed regions to get like whole brain maps of how they relate to the rest of the brain. I'll show examples. And it would be great to have some additional data, external validation, by relating these parcels to some notions of function and cognitive processing. Okay, so let's go into detail. In this example, we have the region of interest in brown. So why are they topographically disjoint? That's because they're two different blobs from two different contrasts from a motor experiment. Okay, so they initially came up in different contrasts, so they should do something else. In this experiment, we just joined these two volume definitions of blobs from a task-based fMRI study into a same region. So the clustering algorithm, and that's often, most of the time the case, is actually blind to the topography of the voxels. Okay, so for most applications of clustering algorithms, the algorithm itself does not know which voxels are neighboring, which ones aren't in 3D space. Okay, there are exceptions. All right. So, and just to give an intuition, let's start here. To give an intuition of how you actually get all this information to distinguish the single voxels, just imagine with these three little dots here. For each of those, which are just exemplary voxels in our region of interest or volume of interest, we get a whole brain map of how each particular voxel talks to the rest of the brain. So now you can imagine if you look at the whole brain maps of two voxels that are very close to each other, they're probably going to be pretty similar. So and that already gives you an indicator. The really kind of like cluster driving property in most of these brain parcellation studies is not the absolute connectivity to the rest of the brain, but the relative differences in whole brain connectivity. Okay? So it's really about how they are relatively different. It's not what the whole brain pattern looks like. All right. So and then this is what it would look like to have Gaussian blurring of these individual foci that various experiments in the BrainMap database featuring at least one focus of activity in the brown region of interest, how they activate in the rest of the brain. Okay? So and across dozens and hundreds of experiments with various different motivations that were published already, you can come up with this map. So this would be, this shows you the co-activation or the co-deactivation with all these voxels combined. So then the final step is usually to draw a statistical inference on the above chance convergence in the brain, in how the whole kind of the rest of the brain statistically significantly talks to kind of the region of interest. All right. Here you see the three separate maps. So although they are kind of close here, it doesn't look exactly the same. So we already went into detail about this. Let's say there are 100 voxels here, and let's say there are 200,000 voxels in the rest of the brain. Then this would be 100, and what you measure is so the properties that you use to do parcellation, to do clustering, which is from this direction here. The properties that you use is how does each of my voxels of interest in brown here, how is it functionally coupled with each of the various other parts of the brain? So now what we haven't said yet is there's also a variety of ways to choose the target voxels. So I think the most common is to just take the various other voxels that are not part of your region of interest, but you could also kind of like give it a more kind of biologically motivated flavor. So for example, you could say, I'm really interested in these sets of brain regions, or I know from monkey literature that anterior cingulate cortex and posterior cingulate cortex and some other regions, they should really be important to my question. It is totally possible to only look at the connectivity to certain target areas here. Okay? So just to put this in perspective, it doesn't need to be connectivity to really every single voxel in the rest of the brain. So that's one. There would be a neurobiological motivation to not choose all voxels in the rest of the brain. You can also sometimes find more signal processing types of motivation. For example, you could perform a principal component analysis in this direction. So instead of having 200,000 properties that describe each of the voxels in your brown seed region of interest, you could just compress it to, let's say, the 100 components that really explain variance across the brain. So, and that is actually often done and gives you a lot of advantages in terms of like computational load, and the result is most of the time essentially identical. All right. We mentioned k-means clustering that is applied, just to emphasize, in this direction. Yeah. So I'm asking these 100 voxels in my brown area, how should they be in a same group or in a different group? So in a same parcel in the brain or not? So again, k-means clustering doesn't know that these two voxels are closer to each other than this one and that one. Okay. And I think we often see these types of plots when k-means is explained in the neuroimaging literature, but I think sometimes it may not be entirely clear what the axes and the points actually mean. So if you just think for a second, yeah? So if you have 200,000 voxels in the rest of the brain, and that's what we use to do brain parcellation using k-means, and we have 100 voxels in our brown region of interest, what are these black dots? I think it's not like obvious to everybody usually, although this is like oftentimes explained and pictured like this. Ideas? Well, there should be exactly 100 dots here, because each dot corresponds to one of these voxels in our brown area of interest. So, and what is this? The axes, what are the axes in this particular example? So it should be the relative difference, right, between the properties that we use to drive the parcellation. So it could look like this if, for example, you performed a principal component analysis in this direction, you keep two axes of variation that capture most variance in your data, then it would look like this. Because in 200,000 voxels in the rest of the brain, you would actually have 200,000 axes, okay? And each of them explains how, like, two particular voxels, so these two here, they would be super similar because their connectivity to this voxel in the rest of the brain and that second voxel somewhere else in the brain is very similar, okay? So this is how it works. So we are in feature space here. All right. What you see in colored here are the centroids. So I'm not going to go too much into detail, but I just try to convey the nature of k-means clustering. So it's an example of an EM algorithm, expectation maximization. So it iterates in two steps that are repeated. So, and it starts with three random kind of like centers of clusters. So you need to pre-assume the number of clusters, as we said. And the locations are random at first. So the first of the two steps then is to assign each of the black dots, so the voxels in our region of interest, to the closest cluster center. Okay? That represents best how the connectivity is relatively different to the rest of the brain. So once we have assigned all the seed voxels, we will recompute the position of the randomly initiated centroid to better match, to be at the Euclidean center of the voxels. And then we'll recompute the center, so we recompute the assignments of the voxels again, okay? So this goes on dozens of times until one of two things usually happen. One of them is the difference in the position of the centroids is super small. So then you kind of converged, yeah, which is not obvious because it's a non-convex algorithm. Or you just reach the maximum number of iterations. So in most applications, k-means clustering, you specify a maximum number of iterations that you go through. And if you reach this, you just choose what you have at that point. Okay, so this is k-means clustering. And the typical question then in this situation is, how do you know the number of clusters? So I think an important take-home message at this point is that we are in a very exploratory, descriptive type of statistical regime here. And this is why in most of these settings, it is very challenging to articulate a null hypothesis of how many clusters do I have. So in most settings, it is challenging to really come up with something like a p-value or null hypothesis test for the number of clusters. So instead, I'll just mention some examples later on. We use cluster validity criteria. So many people know, for example, from principal component analysis, the elbow criterion. But what is important kind of to keep in mind is just because a certain cluster validity criterion, such as the elbow criterion in PCA, just because this itself is an objective way to get an answer, the choice of cluster validity criterion is still a subjective choice. Okay, so even if there's more than 30 cluster validity criteria out there, at least, that you can use to get kind of a quantitative handle on how many clusters do best explain my data. It is ultimately not really possible to reach the same kind of statistical rigor such as in a hypothesis in statistical significance testing. Okay, and that's something that the technical communities have been looking at for several decades, and there's not really a definitive answer for this question. Yeah, so it's the reviewers is a favorite question from the reviewers. Yeah, just in case you're planning a study in this space, you can expect that one or two of the reviewers will ask that, and then you can say it's difficult to give definitive answers in that space. Yeah, and where we go back to what do you ultimately want to say? Yeah, so if you parcellate a region and you have different knowledge, cytoarchitecture, and so on, it's okay to bring this into the equation when thinking about this question. Okay, so here's just some code that tries to show you it may be perhaps less complicated to perform clustering than you think. So what you see here is not really related to the brain, but we are trying to cluster flowers according to 3 properties that are shown here. And so this is the ground truth. This is how these flowers are different in nature. And here you see 3 clusters, 8 clusters, and the assignment of particular data points to 3 or 8 clusters is shown here. So this is an example in Python from a scikit-learn example. And I just wanted to briefly mention that in this case, it's really this line that imports the algorithm itself. And if you already know where your data is and everything, it is this second line, so estimator being one of these k-means clustering, so initialized with 3 or 8 clusters, is this line that does the assignment of the clusters. Okay? So essentially, in 3 lines of code, you have all the information that you need to produce this plot. Yeah? So if you look at this example, I cut it a little bit, but I just wanted to say you can perform a k-means clustering in 3 lines of code. Okay? So it doesn't mean that's everything you have to do. Yeah? There's still a lot of kind of overhead and so on, but it may be perhaps less challenging than you think to perform a type of clustering. All right. There are different types of clustering algorithms. So here's an example from hierarchical clustering that we haven't mentioned yet so much. So it has a different type of flavor. It also doesn't know where a particular voxel is in the brain, but it operates in a completely different fashion from k-means. So it asks in a bottom-up or top-down fashion, so if all of these voxels are completely different in the brain, and I have as many classes as I have voxels in the region I start with, so 100, you would have 100, then in each step you ask, which are the two whose connectivity to the rest of the brain is so similar, is closest, so that I can merge them into a new brain, a new parcel. So in the second step you would have 99. Then it goes on. Okay? So the other direction would be I have only one class in my whole region of interest, and then I start dividing it into two, and so on. Okay? So the important take-home message here is, still in neuroimaging, we oftentimes write model-free approach, hypothesis-free approach, and so on. Each of these clustering algorithms makes some implicit or explicit assumptions about your data, so it is just technically impossible to be 100% model-free oftentimes in practice when you perform brain parcellation. Okay? So that's why you see what I try to kind of picture here. Hierarchical clustering just proceeds in a pretty different way than k-means clustering. And there are situations where you won't get the same answer. And they are driven by the inner workings of what the clustering algorithm does. So we shouldn't be surprised by this. Some of the first things were spectral reordering. So spectral reordering itself is actually not a clustering algorithm. It's just a data transformation to visualize your data. You can use this as a starting point to form clustering, but the early papers didn't actually do that. All right. So to finish up this slide, if you map the assigned groups, yeah, group 1 green, group 2 red, back to the brain, then this is what you get. So it actually does recover the two blobs that you got from the experiment that we merged in the beginning. Okay? So, and then you can, for example, query the BrainMap database that hosts a quarter of the neuroimaging literature that we have today. And you can find that, indeed, this green posterior cluster has many more assignments with action and motion-related types of cognitive processes, whereas this more anterior one, probably in the SMA, supplementary motor area, has much more a kind of higher-level cognition types of assignments, different from motor, and that is working memory, language, cognition, and so on. And if you map it back to the experiment where it actually comes from, you can see that a tapping task and a working memory task, they differently engage these two clusters that we divided in a data-driven fashion. All right. So just briefly skim through kind of some different flavors of what you may ask and what you can do once you have these parcels. Yeah. So oftentimes it's difficult to get a paper published these days if you just say, "Look at my clusters." So you usually need to go beyond to kind of validate them and assign them to function and so on. Yeah. What you can ask, for example, is not only how is my anterior red cluster connected to the rest of the brain, but you can ask how is it statistically more connected to the brain compared to the posterior green cluster. Okay, so something like a functional connectivity contrast. So this is something that is often done. That's what you get. And you can also ask, once you have those, you can ask in which part of the brain do the whole brain connectivity profiles of my red and green cluster actually agree. So this is what you see in yellow, which is more like a whole brain connectivity conjunction analysis. Well, there were a lot of inventions. For example, we can measure how each voxel is connected to the rest of the brain in various ways. So for example, by involving less or more brain map experiments. So then you get multiple maps, whole brain maps per voxel. And then if you average those, you get more robust results that allow you to recover clusters that are close to cytoarchitecture as we know it from neuroanatomical studies. You can also compare across modalities, so the task-dependent and the task-free type of functional connectivity to ask, well, does it really agree, the boundaries that I get, but also the connectivity to the rest of the brain in the two modalities. And it's really a creative process. So what we did here, for example, is we noticed that different literatures about the right temporal parietal junction, they actually have different names and different neuroanatomical descriptions and different processes that they associate to their blobs in the inferior parietal lobe. So we started out by performing meta-analysis in separate literatures that don't really talk to each other that much to come up with 3 statistically significant convergences in 3 different literatures to combine them into a starting region. Okay? So then the question is, of course, do these 3 literatures that don't really talk to each other talk about the same parcels or not? And turns out that in this example we had 2 and not 3 parcels. So there seems to be, or there could be, 2 kind of connectionally coherent entities in the brain that relate to these 3 different types of literature. All right, so I'm going to go through this here. So to summarize a more complete process of running a brain parcellation study, you start out by choosing a region of interest, as I said. There are various ways to do this, and you probably want to align this with what the types of statements that you ultimately want to make. You collect, in the MACM example, brain map studies that activate in that region. You look at how do they co-activate or deactivate with the rest of the brain or particular other target regions in the rest of the brain. You perform a clustering algorithm. You can use heuristics, cluster validity criteria, to get a handle on which choice of cluster number may be more meaningful than another one. Then you typically map it back to the brain, and you can use various types of functional characterization to give sense to the parcels that you have discovered. All right, and there are a lot of different cluster validity criteria, and there's no ultimate definitive answers to how many clusters are best for your type of data set. And I think that's a good finish statement. All right. Questions?